Hello, everyone. Today we're continuing our deep dive of Richard Dawkins and Yan Wong's book, The Ancestor's Tale. In this episode, we're going to discuss nearly all non-animal eukaryotic diversity and Kleiber's Law. So let's jump right in. At this point in our series, I must emphasize that our rundown on the Tree of Life has been heavily biased towards the branches that include us, while the adjacent branches have often received a brief overview. The last 52 episodes were mostly concerned with the animals. There are over 1.2 million described species of animals, over 80% of which are insects, but only 3 episodes were focused on them. Likewise, in this and the following 3 episodes, we will cover all the other eukaryotes, the fungi, plants, and all protists. Last time, we discussed the coenoflagellates, sisters of the animals. Our last common ancestor with them lived between 700 and 900 million years ago during the middle to early Neoproterozoic era. A major recurring issue with molecular clocks, which we discussed at length in the velvet worm's tale, is that fossils are needed to calibrate the dates. Without fossil data, these dates become more suspect the further back you go, which is especially true once we get past the Ediacaran period 635 million years ago. A 2006 paper by Cedric Burney and Jan Pawlowski gives some dates in eukaryote evolution using a handful of well-characterized Proterozoic fossils, such as the red alga Bangiamorpha from 1.2 billion years ago, the yellow-green alga Paleovaucheria from 1 billion years ago, the green alga Proterocladus from 750 million years ago, the fungus Tapania from 1.4 billion years ago, and some testate amoeba fossils from 750 million years ago. The date they give for the common ancestor of animals and coenoflagellates is about 872 million years ago, towards the upper end of that range. The next clade we meet is Philisteria. 2 of the 4 genera in this clade are Ministeria and Capsospora, which have non-tapering tentacles. These tentacles are unaggregated microvilli, which are ultrastructurally similar to the microvilli of coenoflagellates. Indeed, such structures are common across Philisterians, coenoflagellates, and animals, hence the clade uniting them has been called Phylozoa. Ministeria is just a free-living marine heterotroph, and Capsospora is an endosymbiont of some freshwater snails. There are 2 other genera. Pygoraptor is a flagellated eukaryovore, and Zichyspora, described in 2022, is a parasite of amphipod crustaceans. Now we move on to Ichthyosporia, named in reference to the fact that the earliest identified members are parasites of fish. The clade now contains more members, some of which were previously considered to be fungi, and some parasitize other animals besides fish, including humans. Because of this, some have suggested changing the name of the clade to Mesomycetozoa, meaning middle fungus animal. Besides Ichthyosporia, there are a few other genera, Sisymonas and Corallichytrium, which may be in their own clade, called Pluriformia, and are either more closely related to Phylozoa or more closely related to Ichthyosporia. Sisymonas is another flagellated eukaryovore, and Corallichytrium is a roundish protozoan often found in association with coral reefs. Both Pygoraptor and Sisymonas have complex life cycles involving amoeboid and flagellated stages. Together, animals, coenoflagellates, Philisterians, and Ichthyosporians comprise the clade Holozoa, meaning whole animal. In other words, animals and all organisms more closely related to animals than they are to fungi belong in Holozoa. When the common ancestor of this clade lived is based almost entirely on molecular clocks, which, as mentioned previously, can be dubious when not anchored by fossils. Though the Duchanteau Formation from 600 million years ago may hold fossilized microscopic holozoans, the earliest contender of a holozoan is the billion-year-old Bicellum brazieri. This fossil hails from the Scottish Diabag Formation that preserves populations of benthic and planktonic organisms trapped in former lake bottom sediments. Bicellum is a spheroidal mass of nearly spherical cells, called a stereoblast, enclosed by a periphery of sausage-shaped cells. The cells evidently lack cell walls, meaning the organism isn't a plant. Multiple ontogenetic stages of bicellum were found at the site, but they did not show evidence of palintomy or multiple rapid rounds of cell division without intervening growth. Instead of palintomy, bicellum may have grown via aggregative coloniality or the cellularization of a syncytium, i.e., the formation of cell membranes around each nucleus in a single multinucleated cell. However, bicellum is much more morphologically complex than any eukaryotes that exhibit aggregative coloniality, and without cell walls, that just leaves holozoa as the most likely affinity. Next up is a well-known clade, Holomycota, meaning whole fungus. As the name suggests, this clade is the fungal counterpart to holozoa, as fungi and all organisms more closely related to them than they are to animals are included within Holomycota. Combined holozoa and Holomycota form the clade Opisthokonta, almost all of which have a life cycle stage characterized by possessing a single flagellum that pushes the cell from behind. The first clade we meet within Holomycota secondarily lost their flagella. These are the nucleariids, including the amoeboid Fonticula. Fonticula is a slime mold, which is a term that typically refers to a unicellular amoeboid organism that forms multicellular reproductive structures called a saurocarp. Various clades of eukaryotes have independently evolved slime mold lifestyles, making the term informal and polyphyletic. In the case of Fonticula, the reproductive form looks like a tiny volcano. As for Nuclearida, these are all amoeboid and unicellular, using their phylopodia to crawl around. Smaller species like Parvularia consume bacteria, while larger ones, like the namesake Nuclearia, are eukaryovorous. Further differentiating these clades from fungi, they lack chitin in their cell walls, and they have a particular discoidal shape of their mitochondria. A 2020 paper by Mary Burby et al. Argues that fungi were ancestral unicellular, phagotrophic, free-living organisms. They probably resembled flagellated chytrids. From that starting point, fungi diversified into a huge variety of niches. Many are parasites. Candida is responsible for oral thrush and vulvovaginitis. Athlete's foot can be caused by trichophyton, epidermophyton, and microsporum. And Rosella engulfs and digests the cytoplasm of host cells. The vast majority of modern fungi, from chytrids to yeasts to mushrooms, belong to the clade Eumycota, true fungus, which are characterized by possessing cell walls made of chitin and obtaining nutrients via osmotrophy, which, to quote Burby et al., quote, involves the cytoplasmic uptake of nutrients that diffuse across cell walls and often is coupled with the secretion by the fungus of extracellular enzymes that release nutrients from substrates, close quote. Fungi are also predominantly obligate or facultative aerobes that require oxygen to complete their life cycles. Like other eukaryotes, fungi utilize aerobic cellular respiration and require oxygen for sterile biosynthesis. Fungal genomes also encode a number of genes that require oxygen to function, such as lytic polysaccharide monooxygenases for breaking down polysaccharides. There are only two clades of independently obligately anaerobic fungi, Microsporidia and rumen chytrids belonging to the phylum Neocalymastigomycota. I hope you're all happy with that pronunciation. Fungal ancestors evolved in aquatic environments and only transitioned to land with the help of plants. As we saw in our video, Passing Chloroplasts, fungi form symbiotic relationships with both cyanobacteria and plants. Undoubtedly, cyanobacteria were on land before plants and formed intimate associations with fungi, passing products of photosynthesis to them while the fungi passed back nutrients. The evolution and expansion of land plants in the late Ordovician and early Silurian allowed fungi to diversify in turn. The minimum age of 4 eumycete groups, Ascomycota, Glomeromycota, Blastocladiomycota, and Basidiomycota, date to the Silurian and Devonian, anchored by fossils such as Ornatophyllum, Scutellosporides devonicus, and Paleoblastocladia milleri, respectively. Saprotrophic fungi break down cellulose and hemicellulose for energy. Lignin can be degraded by some fungal clades too, but this is only done to expose the cellulose. The fact that cellulose-degrading enzymes are encoded in the genomes of most fungi lends support to fungi processing cellulose and hemicellulose taken from algae for energy, and as the freshwater ancestors of land plants transitioned to a terrestrial existence, fungi trailed them the whole way. Osmotrophy evolved later. When did the common ancestor of fungi and animals live? Recent estimates tend to place it a little over 1 billion years ago. In 2017, Stephen Bankston et al. Reported mycelia-like structures dating to 2.4 billion years ago, but Burby et al. Responded to this by saying, quote, "A suitable source of nutrition for such ancient heterotrophs would be difficult to infer. Furthermore, the deep biosphere is anaerobic, which is inconsistent with the metabolic requirement for oxygen by most fungi. Although intriguing, these filaments do not exhibit any features that would establish a compelling link to fungi, and in our view, even their biological origin remains questionable." Close quote. A more likely early fungus is the billion-year-old Orosphera giraldiae, but these filamentous remains could also belong to algae. And some pseudoseptate filaments attributed to fungi found in the Democratic Republic of Congo date from 1 billion to 800 million years ago. But even if these are fungi, specifying a more precise position for them within the fungal tree of life is simply not possible. Once we get into the Paleozoic, fungal fossils are still rare from the Cambrian to the Silurian, becoming more abundant in the Devonian and onward. As we discussed in the leafcutter's tale, the early Devonian Rhynie Chert formation, dating to 410 million years ago, contains fossils of numerous fungi along with various other organisms, such as arthropods, several groups of green algae, including a charophycean streptophyte alga, oomycetes, testate amoeba, and cyanobacteria. The fossil fungi include saprotrophic, parasitic, and mutualistic species, including the earliest mycorrhizal fungi. For reference, the bodies of most fungi are not mushrooms or puffballs or chanterelles. They are masses of fine root-like structures called mycelia. Each mycelium is composed of threads called hyphae that grow longer and divide into multiple threads. Fungi reproduce sexually when two mycelia come into contact with one another and exchange nuclei. More on this in a bit. One mycelial network may cover acres, as members of the genus Armillaria can. Mycelia may parasitize plants or, in many cases, form mutualistic associations with them. Those mutualistic plant-associated mycelia are called mycorrhiza, and they are especially common in the fungal clade Glomeromycota, though other fungi can exhibit them too. When these mycorrhiza are outside the plant, they are called ectomycorrhiza. When they are inside, they are called endomycorrhiza. Traditionally, there have been 6 recognized groups of fungi: chytrids, zygomycetes, glomeromycetes, ascomycetes, and basidiomycetes. These names refer to each group's life cycle. Chytrids have flagellated spores that look not very dissimilar from the flagellated cells of choanozoans. However, the chytrid stage isn't diagnostic of a particular clade, more likely being the ancestral state for fungi. Some fungi with chytrid stages, such as Blastocladiomycota and Olpidiomycota, are now classed in clades separate from the other chytrids. The same is true for zygomycetes. Zygomycota refers to the fungi having a zygote stage from which the zygosporangia, the spore-producing structure, arises. The zygote forms when mycelia from 2 separate individuals unite. Today, genetics has split zygomycetes into the clade Mucoromyceta and Zoopagomyceta, which are not closely related. Glomeromycota is still recognized, being known for its members mostly being mycorrhizal. And Ascomycota and Basidiomycota are sisters within the clade Dicaria. Dicaria fungi have a unique stage in their sexual reproductive cycle. When mycelia of 2 different mating types meet, the cells of the 2 parents fuse, but the nuclei remain separate within the cell, a process called plasmogamy. Hence why the group is named Dicaria, meaning 2 nuclei. During this 2 nuclei stage, Dicaria fungi develop a fruiting body, which we often refer to as mushrooms. Even at this stage, each cell still contains 2 separate nuclei. These will only fuse when the fruiting body develops spore-producing structures. Here, the fusion of the nuclei, called karyogamy, occurs. The resulting zygotes will immediately undergo meiosis, producing haploid spores. These spread a variety of means, but once they land, they can begin forming a new mycelium. This is in contrast to our reproductive cycle. Animals like us are diploid during most of our life cycle. The gametes, sperm and egg, are the only haploid cells that we produce. But fungi are haploid most of the time, even during the 2 nuclei stage. Zygotes are the only diploid cells in fungi. As we will see later, plants do something different from both fungi and animals. The 2 big groups of Dicaria fungi are named after the types of spore-producing structures: the ascus for ascomycetes or the basidium for basidiomycetes. These also include the most familiar fungi. Ascomycota includes morels, truffles, bakers and brewers' yeast, penicillium and cup fungi, and Basidiomycota includes smuts, rusts, and mushrooms. The most famous fungus from the fossil record is Prototaxites. Often reconstructed as a branchless vertical columnar structure, Prototaxites has been known since 1859, when it was first described by Canadian geologist Sir William Dawson. Growing up to 29 feet, about 8.8 meters, Prototaxites was by far the tallest land-living organism of the late Silurian, and it lived until the end of the Devonian period. Dawson mistook Prototaxites for a rotting conifer, which is why the name translates to first yew. In 1872, British paleobotanist William C. Carruthers challenged Dawson's assignment, arguing that Prototaxites was actually a marine alga. This affinity remained almost unchallenged until 2001, when Francis Huber argued it was a fungus. Huber wrote, quote, "This report has a triple purpose. One, to name as neotype a recognizable specimen collected by Dawson, for which the locality and stratigraphic data are known. Two, to re-describe the genus as structurally composed of three interactive forms of hyphae, i.e. large, thin-walled, septate branching generative hyphae, large, thick-walled non-septate skeletal hyphae, and small, thin-walled, septate branching binding hyphae, which combine to form a gigantic phototrophic amphigenous perennial sporophore with saprobic nutrition. And three, to classify it in the kingdom of fungi." Close quote. In 2007, C. Kevin Boyce et al. Provided further evidence for the fungus hypothesis with carbon isotope ratios that indicated the organism was a heterotroph. Autotrophs make their own food, so we should expect them to fall within a certain range of isotopic values. Heterotrophs, by contrast, would be expected to exhibit a wide range of values because they do not make their own food. Their value should be related to their substrate. Surely enough, this was the case for Prototaxites. In 2022, Vivi Vajda et al. Kept with the fungal affinity, but reinterpreted Prototaxites not as standing, but laying down. Prototaxites, they argued, was a giant rhizomorph, a massive collection of mycelia like those that can be found in modern armillaria. Another paper by Matthew P. Nelson and C. Kevin Boyce argued against either an Agaricomycete sporocarp or Ascomycete lichen association, and for Prototaxites being a member of an extinct fungal clade. Once we leave the fungi, the phylogeny of eukaryotes becomes rather fraught. A recently erected clade is known as Obazoa, in which OBA stands for Opisthokonta, Breviatea, and Apusa monadita, respectively. Apusa monadita is generally considered to be sister to Opisthokonta, and Breviatea is sister to both. Remember that all Opisthokonts are descended from a common ancestor with a single flagellum that pushes from the rear. Evidently, the common ancestor of extant eukaryotes possessed two flagella, a fact which we can infer based on the paraphyletic distribution of double flagella in all the major eukaryotic clades. The Apusamonads are a handful of unicellular bacteriovorous genera with two flagella that glide, and Breviatea contains unicellular amoeboid eukaryotes with an anteriorly oriented apical flagellum. Sister to Obazoa is Amoebozoa, the clade of true amoebas, which are highly varied in their lifestyles. A lot of eukaryotes are amoeboid, which describes how the cell readily changes shape to move, but most of these amoeboid organisms are not true amoebas. For example, the phylum Placozoa, which are animals, the fungal cousins Fonticula and Nuclearida, and the brain-eating amoeba, Naegleria fowleri, which is an excavate. True amoebas may be naked or testate, and as mentioned earlier, testate amoebas, similar to the modern genus Arcella, are known from as far back as 750 million years ago in the Tonian period. They may be uninucleate, binucleate, or multinucleate. Many produce cysts, which themselves are highly morphologically variable. They may reproduce sexually or asexually. They may be sporocarpic, in which a single amoeboid cell differentiates into a usually stalked subaerial structure that supports one to many propagules termed spores, or saurocarpic, in which amoebas aggregate into a multicellular mass that develops into a multicellular fruiting body. They are found in all environments, and amoebas may have two, one, or no flagella. Two flagella are the ancestral condition. In general, amoebas move and consume prey via pseudopodia, which are temporary blobby arm-like extensions. Some of the most well-known amoebas are Amoeba proteus, Entamoeba, and Pelomyxa. The first is a free-living freshwater model organism with an unusually large genome. Entamoeba is a genus known for causing amoebic dysentery, and Pelomyxa is a free-living species that consumes a variety of both bacteria and eukaryotes. Entamoeba and Pelomyxa are anaerobic, having highly reduced mitochondria called mitosomes. Their mitosomes are incapable of cellular respiration, so Pelomyxa relies on symbiotic bacteria for living in aerobic environments. Because researchers initially thought Entamoeba and Pelomyxa lacked mitochondria, they were classed with Microsporidia and the excavate clade Metamonada in the now defunct clade Archezoa. The idea was that these clades share either highly reduced, which was interpreted as the primitive condition, or absent mitochondria. Therefore, they must be early diverging eukaryotes. When Lynn Margulis proposed the endosymbiotic hypothesis, there was no room for such eukaryotes to exist. The mitochondria did not evolve piecemeal from a pre-existing cellular structure. It is itself an originally separate bacterium that was enslaved to produce cellular energy. So, between the strong genetic and cytological evidence Margulis marshaled for endosymbiosis and the burgeoning phylogenetic data on eukaryotes, the Archezoa hypothesis was falsified. The next clade is another acronym, CRUMS, where the C, R, and M refer to Collodictyon, Rigiphylla, and Mantamonas, respectively. Collodictyon is a free-swimming unicellular eukaryote that looks bilobed with 2 or 4 apical flagella. Rigiphylla is a rounded eukaryote that lacks flagella but does have pseudopodia, and Mantamonas is an asymmetric gliding biflagellate eukaryote. Recently, the biflagellate genus Glissandra was added to CRUMS, revealing a morphological commonality among all CRUM members. It appears that all CRUMs have a pellicle, a cytoskeletal layer that supports the cell membrane. And possibly the last group on our side of the eukaryote divide is called Anciromonadida or Planomonadida, which contains the gliding biflagellates Anciromonas and Planomonas. Recent phylogenies have placed the former excavate Malawimonas in this clade too. It's a small, free-living bacteriivore found in fresh water and soil. Now let's recap all of the organisms we've discussed so far. Animalia, Coenoflagellata, Philisteria, Ichthyosporia, Holomycota, Apusamonadida, Breviatea, Amoebozoa, CRUMS, and Anciromonadida. These are collected into the enormous clade Scotocaryotes, also known as Opimoda or Neozoa. This clade is sister to another enormous clade called Diaphoretics, also known as Corticata or Biconta. Biconta doesn't really make sense as a clade name anymore because, as we've mentioned, having 2 flagella is the ancestral state for all modern eukaryotes. There are a few clades that have traditionally been grouped together as excavata, such as Discoba and Metamonada, that were considered the final major clade of eukaryotes, but recent genetic evidence tends to indicate that this isn't a natural group. Instead, the excavates are a paraphyletic or polyphyletic assemblage of eukaryotes. We'll talk more about these in the Mixotrix tale, so ignore them for now. When we turn to the other major clade of eukaryotes, Diaphoretics, we find the tree consists of 2 groups, the clade Archaeplastida and Chromista. As we discussed in passing chloroplasts, the chloroplast is derived from an originally free-living cyanobacterium closely related to the modern species Gliomargarita lithophora. Over 2.5 billion years ago, cyanobacteria evolved oxygenic photosynthesis, and between 2.1 to 1.6 billion years ago, eukaryotes emerged as the product of a mutualistic partnership between an archaean and a bacterium that was ancestral to our mitochondria. After scotocaryotes and diaphoretics split, a second endosymbiotic event occurred in which the ancestor of modern archaeplastids, the red and green algae, obtained the cyanobacterial ancestor of chloroplasts. Archaeplastids entered a complex series of endosymbiotic events following it. Notably, red algae were taken up by cryptophytes, and then those cryptophytes were taken up by the ochrophytes, the clade that includes kelp. From there, the ochrophytes may have been taken up by different clades like Mesozoa, which includes dinoflagellates and apicomplexans like Plasmodium, or the cryptophytes may have been taken up by haptophytes, which includes coccolithophores, with the haptophyte being taken up by Mesozoa. Regardless of the order of transfers, serial endosymbiosis is a common feature of Chromista. Some chromists still have vestigial nuclei in the highly reduced cells of their symbionts, a feature which characterizes the clade Chlorarachniophyceae. which receive their plastids from the green algae clade Viridae plantae. The earliest signs of eukaryotes in the fossil record are the acritarchs. Recall that acritarch is a wastebasket category for probable eukaryote fossils that have not been inserted into a specific clade. The earliest date to between 2.15 to 1.95 billion years ago, and the earliest identifiable diaphoretics are the red algae Raphatasmia and Ramathalis from 1.6 billion years ago. In the sponge's tail, we discussed the transition from a cyanobacteria to an archaeplastid-dominated world during the Cryogenian period 720 to 635 million years ago, the evidence for which comes from differing levels of certain steranes. However, the famous phytoplankton, such as diatoms, dinoflagellates, and coccolithophorids, diversified much more recently, 250 million years ago at the start of the Triassic period. The expansion of armored phytoplankton in the Mesozoic era has been proposed to have resulted from the breakup of Pangaea, providing newly flooded continental margins with high nutrient habitats. Since rhodophytes appear so early in the eukaryotic fossil record, that means excavata, scotokaryotes, chromista, and archaeplastida had already split by 1.6 billion years ago, some 300 to 400 million years after eukarya arose. The rapid diversification following the origin of the clade is likely the reason early eukaryotic splits are so difficult to determine. Some recent work has been done to pin down some enigmatic protozoans, such as Hemimastigophora and Provora. These are clades of predatory eukaryotes whose placement on the tree of life has only just been elucidated. They may be sister to all other members of diaphoretics. Chromista is split into several clades. There are the SAR groups, Straminopiles or Heteroconta, Alveolata and Ritzaria, Telenemia, and Haptista. Straminopiles are biflagellates with one smooth flagellum and one covered with hairs. The most well-known members of the clade include diatoms, the silica-shelled eukaryotes that make up a substantial portion of phytoplankton, and oomycetes, which were previously mistaken for fungi. Although oomycetes are often called water molds, they may parasitize aquatic or terrestrial species. One infamous oomycete is Phytophora infestans, which causes late blight, the plant disease responsible for the Great Irish Famine in the 19th century, which killed about 1 million people. One clade of Straminopiles has independently attained multicellularity called Phaeophyce, which includes kelp. This clade is very diverse in morphology, from members just a few cells in length to forests up to 150 feet tall. A major innovation of phaeophytes is the haplodiplotic or sporic life cycle. This requires a bit of explanation. Earlier in this video, we mentioned that animals are diploid during most of their life cycle, and only diploid cells undergo mitosis, while the gametes are the only haploid cells which never undergo mitosis. We refer to this as a diplontic life cycle. On the other extreme, organisms like the aforementioned fungi are haploid for most of their life. Only haploid cells undergo mitosis, and the only diploid cells are zygotes, which immediately undergo meiosis after fertilization. This is a haplontic life cycle, which is very common among other protozoans as well. In between these extremes is the haplodiplontic life cycle, where mitosis occurs in both the haploid and diploid phases, called the gametophyte and sporophyte, respectively. Multicellular algae, including land plants, exhibit this type of life cycle. To understand this, let's start with the haploid gametes that fuse to form a zygote. That zygote is the start of the diploid sporophyte, which is so called because it ultimately produces spores by meiosis. The sporophyte grows in size and generates spore-producing structures, i.e. the sporangia. Those haploid spores are released to the wind or water, and when they settle, they grow into a haploid gametophyte that will eventually produce gametes. The switching back and forth between a sporophyte and gametophyte is called alternation of generations. With regard to plants, either the gametophyte or the sporophyte is the dominant stage, but this depends on the group in question. The haploid gametophyte is dominant for mosses, hornworts, and liverworts, while sporophyte-dominated life cycles are seen in lycopods, ferns, gymnosperms, and angiosperms. Early vascular plants from the late Silurian and early Devonian, like Aglaophyton, Rhynia, and Cooksonia, are intermediate, having gametophytes that are smaller than extant non-vascular plants, but larger than extant vascular plants. More on plant evolution in a bit. The next clade, Alveolata, includes Dinoflagellata, Apicomplexa, and Ciliata. Dinoflagellates are mixotrophic unicellular eukaryotes, meaning they can consume prey or photosynthesize using their red algae endosymbiont. While they mostly constitute a portion of marine or freshwater phytoplankton, some dinoflagellates are parasites, such as Oodinium that causes fish velvet disease, and Carenia brevis that causes red tide. Others are less harmful and quite beautiful. Noctiluca is able to bioluminesce, producing a bright blue glow. You may have seen these glows during a sunset at the beach where the waves are breaking at the shore. We met a dinoflagellate previously in the polypifer's tail, the symbiotic Symbiodinium, that performs photosynthesis within coral and anemone tissues. The apicomplexans, by contrast, have reduced their chloroplasts to just a vestige of its former self, the apicoplast, which isn't capable of photosynthesis anymore. In becoming parasitic, apicomplexans have evolved complex, multi-stage life cycles. Well-known members of this clade include Plasmodium, the parasite responsible for malaria, which kills around 500,000 people every year, and Toxoplasma, which causes toxoplasmosis. Effects of this disease can range from flu-like symptoms to neuropsychiatric disorders. As for Ciliata, this clade is commonly known as just the ciliates because its members are covered with, you guessed it, cilia. Some 4,500 species of ciliates have been described from marine, freshwater, and terrestrial environments. Though many are free-living heterotrophs, some are parasites, and others are endosymbionts. Some famous members of this clade include Paramecium, Stentor, and Vorticella, the last of which has fossils dating back to the late Triassic period, some 200 million years ago. One unusual feature of this clade is that their cells have at least one micronucleus and macronucleus. The micronucleus is normally diploid and functions only as the germline. Ciliates vary in how many micronuclei they have. Tetrahymena has just one, Paramecium may have two or more, Oxytricha have 2 to 4, etc. All these micronuclei are genetically identical, forming through micronuclear mitosis. Meanwhile, the much larger macronucleus is involved in asexual reproduction and the day-to-day functioning of the cell. Again, ciliates vary in how many macronuclei they have, whether 1 in Tetrahymena and Paramecium, 2 in Oxytricha, or hundreds in Urostyla grandis. When cellular division occurs, the multiple macronuclei fuse into a single composite nucleus that then divides its DNA into the appropriate number for each daughter cell. When sexual reproduction occurs, the diploid micronuclei undergo meiosis to produce 4 haploid micronuclei. A micronucleus is exchanged between individuals, which then fuse to create a new diploid micronucleus, and some of these micronuclei grow into macronuclei. Fascinatingly, some ciliates, like the subclass Hypotrichia, have actually re-evolved sex by cellular fusion. The third of the SAR clades is Ritzaria, which includes the chlorarachniophytes mentioned earlier, as well as foraminifera and radiolaria. Foraminifera and radiolarians are shelled protozoans. Most forams have calcium carbonate shells, while radiolarians have silica shells like diatoms. These two have internalized red algae at some point in their evolutionary history, which is why they can photosynthesize. Forams and radiolarians appeared right at the start of the Cambrian and have an extraordinary fossil record, documenting even speciation events. For this reason, these and the other shelly phytoplankton are important index fossils, meaning that they have a wide biogeographic distribution for a particular period of Earth's history. That allows paleontologists to know with confidence the age of a certain stratum by the existence of fossils of a particular species. In fact, paleontologists use index fossils to define different epochs, especially within the Phanerozoic eon. Closely related to the SAR clade is Telenemia, a clade of unicellular, free-living flagellates found worldwide in freshwater and marine environments. Haptista is the clade that includes coccolithophores and centrohelids. Centrohelids are spherical protozoans with projecting axopods. These are semi-permanent pseudopodia that internally have a complex array of microtubules. On the other hand, coccolithophores are important phytoplankton that possess an exoskeleton made of calcareous plates called coccoliths. Because of this feature, they, like diatoms, foraminifera, and radiolarians, fossilize extremely well, having a record that goes back to the late Triassic, and they too function as index fossils. As it happens, the fossil record of coccolithophores is easy to see. They make up chalk deposits like the White Cliffs of Dover. The Cretaceous period is named for its chalk deposits. During that period, coccolithophores underwent a series of evolutionary radiations, diversifying extensively until the Cretaceous Paleogene extinction wiped out over 90% of coccolithophore species. Often associated with Haptista is Cryptista, a clade of autotrophs and mixotrophs that includes Rhodomonas and Cryptomonas. A distinguishing feature of cryptophytes is the ejectosome, an organelle that contains two spiral threads held under tension. When the cryptophyte becomes irritated, the ejectosome releases the thread, propelling the cell away. However, a 2015 paper by Thomas Cavalier-Smith et al. Found that Cryptista was sister to Archaeplastida, the clade of Glaucophyta, Rhodophyta, and Viridiplantae. Archaeplastida means old plastid, referring to the fact that the common ancestor of this lineage attained plastids first. The exact topology of this clade has long been disputed, which is likely due to incomplete lineage sorting and small lineage divergence times, the usual suspects. Glaucophyta is a small clade of unicellular algae known to inhabit both moist terrestrial and freshwater environments. They may be the outgroup to Rhodophyta and Viridiplantae. Next, Rhodophyta is the red algae, which encompasses both unicellular species like Cyanidiophysce and multicellular ones, which are pretty much all the rest. They inhabit marine environments with a small fraction living in freshwater. Rhodophytes get their red color from the plastid pigment phycoerythrin, and similar to kelp and green algae, rhodophytes have a haplodiplontic life cycle. Their life cycle, though, has an odd twist. It's triphasic rather than biphasic. Kelp and green algae go through a haploid gametophyte phase and a diploid sporophyte phase, but red algae go through a haploid gametophyte phase, a diploid carposporophyte phase, and then a diploid tetrasporophyte phase. Much like in green algae, the gametophyte produces haploid gametes, but when a motile sperm cell contacts the sessile ovum, a diploid carposporophyte forms. The carposporophyte generates diploid carpospores by mitosis, which float off to settle as the tetrasporophyte. The tetrasporophyte produces tetraspores by meiosis that float off to become male or female gametophytes. Since rhodophytes retain the ovum on the gametophyte, they have their own equivalent of a pollinator, a fertilizer to be more precise, such as the isopod Idatea baltica that transports the flagellum-less sperm. The third archaeplastid clade is Viridiplantae, the green algae. This clade splits further into Chlorophyta and Streptophyta, whom together have enormous morphological diversity. There are unicellular and multicellular species, uninucleate and multinucleate individuals, spherical and star-shaped species like Volvox and Microsterius respectively, sheets like Ulva, siphon-shaped species like Chara, one of the largest unicellular organisms known, the 2-inch-long Valonia ventricosa, and of course, the land plants. Chlorophyta contains several clades. There are the planktonic marine and freshwater prasinophytes, which constitute a paraphyletic assemblage, chlorophyceae that can live in freshwater or terrestrial environments, such as Chlamydomonas and Odogonium, and Trabuxiophyceae and Ulvophyceae that can live in marine, freshwater, or terrestrial environments. The majority of phycobionts within lichens are Trabuxiophyceae members, such as the recently described Volcanochloris. And there are many well-known Ulvophyceae, including Caulerpa, Cladophora, Acetabularia, and the namesake sea lettuce, ulva. The oldest known green algae is currently an ulvophyte called Proterocladus that dates from 750 million to 1 billion years ago. However, as we've seen, eukaryotic diversity was low until after the first cryogenian glaciation. Once in the Ediacaran period, oceanic oxygen content rose as algae diversified, and as metazoan grazers evolved increasingly complex feeding strategies, this triggered an evolutionary arms race between them and algae, resulting in algae developing defenses such as larger sizes and mineralized skeletons. Evolutionary arms races may have occurred among algae too, such as for benthic space, access to light, means of increasing nutrient uptake from the water column, and means of spreading gametes. This, in turn, may have resulted in more efficient energy transfer, richer food webs, and the modified shallow marine ecosystems, which may have allowed the evolution of larger and more complex animals. The clade that includes land plants is sister to Chlorophyta, called Streptophyta. Land plants, the clade Embryophyta, nest within a paraphyletic assemblage of algae called the charophytes. This assemblage is named for chara, also called the stonewort, that grows in fresh water rich in dissolved calcium. Chara is covered in calcium carbonate, and when it accumulates over geologic time, it, along with organisms like ostracods and foraminifera, forms limestone. Curiously, the life cycle of chara is quite different from that of land plants. Chara is haplontic. It only undergoes mitosis when haploid. All land plants, by contrast, are sporic. However, as mentioned earlier, the evolution of land plants has involved progressively greater elaboration of the sporophyte phase. Most of the life cycle of mosses, liverworts, and hornworts occurs in the haploid gametophyte phase, and the gametophyte of lycopods and ferns can exist without the sporophyte, though the gametophyte is much reduced. In the late Devonian, this dynamic switched. The evolution of seed plants, the clade Spermatophyta, involved continued reduction of the gametophyte until there were no more than a few mitotic divisions. Like many other groups we've covered, the land plants are preceded by fossils that bear some of their characteristics. Parafunaria sinensis was discovered in Chinese strata dating to 520 million years ago, the early Middle Cambrian, and bears some characteristics of later bryophytes, including a complex rhizoid, capsule, and seta. The earliest evidence of terrestrial land plants comes from late Ordovician spores, not especially dissimilar from modern non-vascular plants. They are called non-vascular plants because they lack the xylem and phloem tissues of vascular plants. There is debate over the phylogenetic position of mosses, which are Bryophyta sensu stricto, hornworts, which are Anthoceratophyta, and liverworts, which are Hepatophyta. Some phylogenies place them together as a single monophyletic clade sister to all other land plants, while others place them as successively more derived embryophyte clades. By the late Silurian, some 428 million years ago, lycopods, which includes club mosses, quillworts, and gigantic extinct tree-like forms such as Lepidodendron, and ferns had appeared. A paraphyletic grouping of tree-like plants called progymnosperms arose in the Middle Devonian, which confusingly was ancestral to both gymnosperms and angiosperms. One of the most well-known of this group was Archaeopteryx, not the stem bird Archaeopteryx. Archaeopteryx acts as an index fossil denoting the time period from 382 to 323 million years ago, the late Devonian to the Middle Carboniferous. Progymnosperms went extinct in the Permian-Triassic mass extinction. Molecular clocks indicate that gymnosperms, the cone-bearing plants, and angiosperms, the flowering plants, diverged from each other more than 300 million years ago. We'll talk about the evolution of gymnosperms in the redwood's tale, so let's move past them for now. That brings us at long last to the angiosperms. Angiosperms earned the moniker of abominable mystery by Charles Darwin due to their shrouded origins, and indeed many questions still remain. The oldest unambiguous evidence of angiosperms comes from pollen from Israel, Morocco, and southern England, dating to 135 million years ago, the early Cretaceous. However, locating stem angiosperms has been difficult. A 2018 paper by Charles P. Scutt identified Glossopteridales and Pentaxylon, Bennetettales, and Catonia as progressively more derived stem angiosperms, helping to fill in some of the gap. Within the angiosperms, there are the so-called basal angiosperms, or the anagrade angiosperms, which are Ambarella, Nymphaeales, and Austrobaleales, magnoliids and their kin, monocots, and eudicots. The plant that tells the tale today is the cauliflower, a cultivar of Brassica oleracea, which is in the eudicot family Brassicaceae. So now, let's discuss the cauliflower's tale. Remember way back to the handyman's tale that there is a general trend for brain-to-body size ratio for vertebrates, called the encephalization quotient, or EQ, that was coined by American biologist Harry Jarrison in 1973. The idea involved scaling brain-to-body size to a single proportional metric on a log-log graph, allowing Jarrison to conclude that the slope of the line is, rather specifically, 3/4. In other words, for every 3 units you go up on the graph, you go 4 units right. If you remember those aspects of the handyman's tale, then you also probably remember that EQ suffers from some theoretical and practical problems. However, those aren't really relevant right now. Curiously, EQ isn't the only biological trend that is represented by a 3/4 slope. The correlation of body size and metabolic rates does too. In 1932, Swiss biologist Max Kleiber compared the body sizes and metabolic rates of a variety of homeotherms, i.e., animals that maintain a stable internal temperature, finding this three-fourths slope and calling it the Kleiber law. Kleiber's work disproved earlier speculations that metabolism should follow a two-thirds slope based on some simple geometry. As organism size increases, their volume, and therefore mass, increases at a much faster rate than their surface area. The researchers envisioned that the slope should reflect selection against producing more heat than can be lost by the organism. Understand that bodies lose heat via their surface area, and more heat is produced by their mass. Therefore, the slope should be two-thirds to counteract the square-cube law, which states when an object undergoes a proportional increase in size, its new surface area is proportional to the square of the multiplier, and its new volume is proportional to the cube of the multiplier. Kleiber's experimental work, however, showed that the slope is three-fourths, not two-thirds. Alternatively, raising body mass to the power of three-fourths gives the metabolic rate. Perhaps surprisingly, this slope doesn't just hold for homeotherms like birds and mammals. It also holds for poikilotherms, or animals that can't regulate their internal temperatures. It even holds for unicellular protozoans and bacteria. However, while the slope for each group, homeotherms, poikilotherms, and unicellular organisms, is identical, their y-intercept is different. The y-intercept is highest for homeotherms, lower for poikilotherms, and lowest for plants and unicellular organisms. The question then is, why? Why does the Kleiber law exist? One answer would be put forth in 1997 by physicist Jeffrey B. West and biologists Brian J. Inquist and James H. Brown, hereafter Webb. Their paper, A General Model for the Origin of Allometric Scaling Laws in Biology, puts the question in the context of how essential materials are transported throughout the body. For very tiny organisms, oxygen and other materials can enter the cells directly by crossing the cell membrane, and cell wall if they have one, but larger organisms cannot do this. Think about trying to eat by merely placing your hand on food. Likewise, we can't breathe by exchanging oxygen across our epidermis. We inhale through the nose and mouth, carrying the oxygen down to our lungs. The lungs branch fractally into bronchi, bronchioles, and then the alveoli that exchange gases directly with the blood. Fish also breathe with their specialized respiratory surfaces called gills, and insects with their system of trachea. Similarly, we move materials within our body via the circulatory system, and plants too have tissues for conducting materials up and down their stems. The first principle of the Webb model is that the nutrient supplying network is always arranged in a fractal pattern. In other words, the pattern is identical regardless of scale. Cauliflowers exhibit very easy to see fractal patterns. Just purchase one from the grocery store and cut it sagittally, so they get the tail. The Webb model suggests that the difficulty of transporting materials places a limit on the ideal metabolic rate, giving that three-fourths slope rather than a slope of 1. The other principles of the Webb model are that the transport system occupies some space in the body, obviously, and that the final branch of the network, in the case of our circulatory system, the capillary, is the same size regardless of the organism, whether human or mouse or mushroom or redwood. It even applies to the cytoskeleton of unicellular organisms. However, towards the tip of the network, the branch occupies a substantial amount of space. If you double the number of cells that need materials, then the volume of branches more than doubles because a new, larger branch is needed to service the smaller branches. If, on the other hand, you wanted to double the number of supplied cells while only doubling the volume of the branches, you would need a much more sparsely laid out network. The third point is that the percentage of an organism's body occupied by the fractal network is about the same regardless of the size of the organism. For mammals, the circulatory system takes up between 6 to 7% of the total body volume. This is done to minimize the energy dissipated in transporting materials. So again, if we want to increase body size and therefore cell number without massively increasing the body volume taken up by the network, then the network must be sparsely laid out. However, a sparser network means less materials are supplied per cell. Thus, the metabolic rate must go down, and the line reflecting log metabolic rate compared to log body size has a slope of 3/4. Now, more recently, some researchers have argued that the Webb model fails to explain the Kleiber law at all, while other elaborations on the Webb model have relaxed the assumption of a fractal network, finding that the models work even if it doesn't require all its original assumptions. Other researchers have found that the slope doesn't exactly match 3/4, but sometimes that it does trend towards 2/3 instead. At any rate, the Webb model is one attempt to explain the Kleiber law, the relationship between body size and metabolic rate. So, thanks for watching, and we'll see you all next time.